stage after the meeting. So Dr. Nadia Roque is a professor at Federal University of Bahia, Universidade Federal da Bahia, in Salvador, in Brazil. She's one of the leading experts in composite in Brazil. It's an honor to have her here today. She was one of the people responsible to organize all the team that composed the Asteraceae treatment for Flora of Brazil, which is an amazing feature in itself. She's been studying Gokinachi and composite in general since her PhD. And she has worked with Dr. Vicky Funk, with Morgan Gossel, who's going to give the next talk in the session, and she's been collaborating with a lot of people in Brazil and outside Brazil. So we're very happy to have her here today. And she's going to give an introduction of the Gokinachi tribe and the main morphological features of this very interesting tribe that is sometimes forgotten. And as you can see, pets are very welcome in this talk. We have special participation from Morgan's dog and Jennifer's cat. Hi. And if you and Rio is in the chat too. So if you have a pet, you can turn on your video and show us our pet sometime. We are very happy to see them. So Nadia, you can take over now, and please keep your mics muted during the talks.
Hi, Carol. Hi, everyone. Thank you so much for inviting me. It's a pleasure to stay here, see my colleagues, my students, and I hope I used to talk fast in Portuguese. So I try to talk slowly to make me understand, and that's it. Okay, so I will talk about the tribe Gokinachi. And I decided to explain or make an overview of the classification and diversity of the tribe. Oops, right here. I will start to talk, to mention this, for me, the main work, like a milestone to the subfamilial classification of composite. It was the first Paneiro and Funky 2002. In this work, the author developed a molecular phylogeny based in several molecular markers in that time. And they produced a phylogeny based mostly in Mutis sensulato and the circumscription of the subfamily Sicoroideae. The results from this paper proposed 5 new subfamilies in red colors and 7 new tribes, including Gokinachoideae, it's here in the arrow, and Gokinachoideae with only 1 tribe, Gokinachi. Look at the basal grade of the family is lots of new tribes they are trying to understand. Well, in this paper, Gokinachi, the tribe, was composed by 5 genera restricted to America. Gokinotaminus, 2 species, Cyclolaps, 1 species, Gokinacha, the major genera, and I think the most complex genera in the tribe. Richterago, that time 70 species, and possibly Shukoa, 1 species. Here we have some representatives of this genre. Well, the authors described the tribe based on Cabrera, Hansen, and Bremer, and Freire that supported the Gokinachi, the tribe, by having an apiculate anther-appendage. Can you see that? Here, its appendage is apiculate. So these authors considered the group of genera that all of them possessing this kind of appendage. So Paneiro Funk decided to consider this character as important. Here, having a drawing, can you see the character? And here is again the apiculate appendage, anther-appendage. Well, what's the difference among the other tribes considering this character? We can see, for example, the Stifti and the tribes, the Stifti and Wunderliche, you can see the appendage is acute and not apiculate. So it's different. It's similar, you can say, but so teeny the differences, but it's real, and you will discuss about it in the talk. And the second character, the Paneri funk used to corroborate the tribe, is the style branches. The style branches in Gokinachi is shortly bilobed, rounded, smooth, and dorsally glabrous. This is very, very important to define Gokinachi. So these two characters recognize the tribe. But we have short bilobed style branches in other groups of basal lineage of compost. For example, you can see here, this is a tribe Bernadesi. This drawing was made by Cabrera, and you see all of the genus has a short bilobed as Gokinatoid, but you can see they are not smooth or glabrous. You can see the papillus below, or the apex or below the bifurcation. In Foucaulder is the only type where the style is swollen below bifurcation. So it's different. It's short, bilobed, but it's completely different from Gokinatoid. Here we have another tribe that is located in the basal lineage. Wunderlichioid is short also, but you can see there are papillus below the bifurcation. So it's different. That one, both characters, the apiculate appendage, anterior appendage, and the style branches, rounded and glabrous, is very good to understand the Gokinachi. However, we have other 4 genera that present the same characters. Hyales ianthopapus from South America and Leucomeris inuele from Asia. Maybe these same characters in this genera probably gave support to the grouping of this genera in a Gokinacha complex by Freire in 2002. Here we can see the diagram proposed by Freire, and it was the first work that they used the style branches added with the anterappendage apiculate. So it's a very important work. Then what the group described 8 sections of Gokinacha, it here. And here we have the genus from South America and Asia that is peripheral of the main group. In 1908, Paneri Funk, in a deeper molecular phylogeny, recovered Gokinatoid with 1 tribe, Gokinacha, as monophyletic and well supported. Clade comprised now 4 genera, not 5. Gokinacha, the major one and more important, Chinoctaminus, Cyclolaps, and Richtera. Why Shukoa? Where is Shukoa? Shukoa, following, according to Freire, it was not, should be excluded from the Gokinacha because the style branches are pilose, dorsally pilose. So it was excluded from the Gokinacha. In the 4 genera that share the same characters with Gokinacha, they appeared in a new tribe, Hyalidae, here. So we can see both Gokinacha, Gokinatoid, Gokinacha, and Hyalidae share the same characters, or the main characters used to corroborate these 2 tribes. So to contribute to our understanding, of character evolution of the family, Vic Funk and I started to study the morphology of the main groups of the basal clades. So our intention was investigate potential new or reevaluated morphological characters in the basal lineage of the compost. we use it as base the phylogeny proposed by Paneri Funk 2008. And we, in this time, I spent I spent a few, I think 3 months in US herbarium, studying all groups or all genera from the basal clade. Well, in the first analysis, we can see these characters occurring in Iality and Gokinacha probably evolved independently for them. So new morphological elements should be evaluated. And I have a good experience in my PhD with the pappus. The pappus until now wasn't analyzed as a good feature to add to in this group. Although most representative of basal clades have pappus of bristles, can you see that bristles, barbellate bristles? We observed a variation of number of series and elements in the pappus. But when did the pappus features become relevant in the taxonomy of these basal groups? My insight to look at the pappus, it was not mine. I was doing my PhD in Kew Gardens when I took access to this identification key from Lessing, a genius Lessing. And I was looking for this identification key when I saw two steps, pappus uniseriate and pappus bimultiseriate. I didn't understand because Ceres, it's here in an uniseriate, and Gokinacha, it's here in bimultiseriate, but they are belonging, sensu Cabrera, in the same genus. So after that, I saw the Lessing paper, I decided to study all the pappus in the basal clades. So it's not my insight, but his insight. And everything changed after I follow Lessing, how can I say, analysis. Well, studying these species in general, we found that Stifte, Wunderliche, and Iality, the pappus has 3, 4 to 5 series and a big number of bristles, and Cypsela 10 ribbed. Can you see it in this image from Wunderliche? You can see the 5 series of the bristles. And the number is much bigger when you compare to Gokinacha. And Gokinacha, we have 2 or 3 series. You can see 3 series, and the number of the elements is lesser, is not too high. And the Cypsela is 5 ribbed. So we proposed it in Hockey Funky 2013, we proposed to add the pappus features to the other 2 characters, like anter appendage or style branches. So we have three features that can help to describe all the genera and species in Gokinachoides. Well, after that, we have understood more the Gokinachi, but inside the tribe, the position was not, the several groups was not strongly supported. So Funky collaborators produced a phylogeny of the tribe, including over 60% of species, all the genera and all sections of Gokinacha. Remember, we had 8 sections from Gokinacha to examine evolutionary relationships among the taxa. This work was very, very important because recognized 8 lineages and suggested a non-monophyletic Gokinacha. So Gokinacha was split in this Mochiniastrum, Anastraphia, Gokinacha from Mexico, Gokinacha senstrito, Pentaphorus. We have two others genera, Quinocutaminus and Histerago, and Cyclolepis is maintained as insecticides. So this is a very resolute phylogeny resoluted in Gokinacha. Lastly, Funk and collaborators published a new genus to this species from Mexico and United States. So we have this format of Gokinacha. In 2019, very recently, Paneiro produced a new phylogenetic analysis to elucidate the taxonomic limits of the genus Gokinacha, and the results showed that Nahuatl, recently described by Funk and collaborators, was not monophyletic. It appears in 3 different clades. So he decided to propose a new genus to Tehuasca magna, a new genus to this species before Nahuatl magna, to Tehuasca magna as a sister group of Anastraphia. So we have a new genus for the tribe. And the last one is a new genus proposed by one species, Gokinacha rotundifolia. This species is very interesting because the collections are very old. And this is the period we have collections. I think because a few in all the collections, the extraction of DNA is unsuccessful. So this species couldn't stay or be included in the Funk in this paper. However, due to distinct morphology, Roque and Sancho provided the evidence that support Gokinacha rotundifolia as a sole species in a new genus. This new species, new genus, was named in honor to Vic Funk, and the genus is Vicia. Here we have, according to Maxima Parsimony, we can show the Gokinacha rotundifolia is within, nested in Gokinacha. Here we can see the plate of the species. We can see the main characters used to distinguish or describe Gokinacha. So this set of features is not found in any tribe of the family older than Gokinacha. So we are sure it belongs to Gokinacha. Vicia rotundifolia is distinguished by actinodromous basal leaf venation with 3 primary vines, very different for all the groups of the tribe. Here we have actinodromous suprabasal leaf in Pentaphorus and Gokinacha, and most of the genera presents pinnate leaf venation, Anastraphia, Nahuatl, Quinocutaminus, Moquiniastrum, and Histerago. So the venation, the leaf venation of Vicia is very strong to corroborate it's a new species. Currently, we have 10 genera to Gokinacha, 10. So below, we can see all the papers that worked with this group and genera, all of them is here. So we have Histerago, is an endemic genus to Brazil. Quinocutaminus occur in Bolivia, Argentina. Anastraphia is endemic to Caribbean area. Gokinacha from Mexico and United States now, we have 2 genera, Nahuatl and Tehuasca. This group, Gokinacha and Pentaphorus, is from South and Central Andes. Moquiniastrum is widely distributed in South America. And here, Vicia is a new one, and Cyclolepis maintains as insecticides. Well, I would like to present for you this 10 genera, And I decided, how can I do that? I would like to make like a identification key to make it easier to understand or all the morphology. So I remember Lessing, and I decided to use as the main character to divide, to share the genre, the Pappus features. So I use Lessing and uniseriate versus 2-3 seriated pappus. Here, just to know, again, the pappus uniseriate and all the bristles are equal in length. And here, pappus 2-3 seriate and the bristles are unequal in length. So this is the two main characters I use as to start our identification key. In bold are autapomorphic characters for each genus. Well, we start with Anastraphia, 33 species endemic to Caribbean. These species, Pappus uniseriate bristles, we have just in 2 genera. One of them is Anastraphia. The main character, main diagnostic character usage for the genus is the lamina with spinose dentate margins, or at least in juvenile leaves. However, we have species without this without margin spinose. But when I was studying these species in US herbarium, I observed the corolla tube shorter than the lobes, and we cannot see any other genera in Gokinachi. Besides, I saw Pappus biseriate on the corners or the cypsela. The Pappus is uniseriate, but in the corners, you have maybe a rib, and you can find more it's maybe 2 bristles. So these 2 characters can be should be analyzed in the future to corroborate or not if it's a taxonomic importance for the genus. The other genus is have Pappus uniseriate is Richterago. Richterago has 16 species endemic to Brazil, and most species are herbs, rosulate herbs, like you can see here. And besides this habit, we have the life form. The life form is Hemicryptophytes. You can see the apical bud in the soil level, and you have the rhizophora and the adventitious roots. So this kind of habit and life form is occurring only in this genus. We have other life forms like geophyte. Can you see that? It's a herb. It's a herb the same way, but the apical bud is, how can I say, is below the soil level, and the life form is geophyte. And the next one is subshrubs. The subshrubs occur in Richterago, but the life form is Camephyta. So herbs and subshrubs is unique in Richterago. In any other genera of Gokinachi, you can find this kind of habits. Richterago is so different because there are species with radiate heads, with the marginal are pistillate, and in the disc we have bisexual flowers, and another species you have discoid heads, but all the flowers are bisexual. So it's the only genus that occurs these 2 different heads. Well, after that, all of the genera all of 8 genera remaining possess Pappus 2-3 seriate. One of them is a Quinicotamnus, found, there are only 2 species found in Bolivia and northwestern Argentina, and it's the only genus that the species are radiate and homogamous heads. or it means all flowers are bisexual, is the only in the tribe. The another one, after that you have other 7 genera, the last 7 genera, all of them have discoid heads. The Cyclolaps, there are only one species, Cyclolaps genistoides, Paraguay and northern Patagonia area of Argentina. And the diagnostic character we can see it's the shrubs with short branches modifying thorns. Can you see that? And leaves are deciduous. So it's a very it's unique in the Gokinachi. The next one is Pentaphorus, 2 species, Argentina, Chile. The diagnostic character is the leaves usually sticky and is distinctly pockmarked and glandular punctate. It occurs only in this genus. The next one is Gokinacha from Central Andes with the 10 species. We can understand or we can recognize the species by heads, usually solitary, but you can find 3 at apex, subintended by 1 to leaf bracts. This genus is it's needed to be revisited, to be studied in the future, because we don't know too much about this species. So we have, it's necessary a revision, deeper revision of this genus. Nahuatl, before 7 species, now 6 species. We can recognize these species because the heads used to be in clusters of 5 until 20 heads. And it's interesting because the heads, usually the bracts come down in the peduncle. So it's very common, peduncles commonly densely scale bracted. It's in this Nahuatl. According to Panero, Terhuasca magna differs from Nahuatl because he found some glandular trichomes on corolla lobes in Terhuasca and no glandular trichomes in Nahuatl. I don't know to talk about it, but in Brazil, it's very common, these glands appear and disappear. It sometimes depends on the habitat, where is the plant. They have to know the habitat or the place when the species are. So I cannot say too much about this character, but that's it proposed by Panini. And not finally, but we have Vicia. Vicia is a shrub. Unfortunately, the last collection was in 1965. No other specimen in this species has been collected in the field. We are trying to find in few patches of savanna in São Paulo, but we couldn't find it yet. And I told you about the venation. It's uncommon, but I think it's interesting to say that I think 99.9% of Gokinacha species has discolored leaves. The abaxial face is cinereals, argenteals, or cream, but it's discolors. But Vicia is concolors and glabrous, and it's not so common in the tribe. And the venation is completely unique. Well, at least we have Moquiniastro. Moquiniastro with the 22 species. Look at Brazil. Brazil, we have 20 species occurring in Brazil. It's a very important place to the genus. Moquiniastrum are trees or mostly shrubs. And the capsulescence is paniculiform, like here, or corymbiform, like here. Leaf or bracteolate. I'm not sure, but I think these characters, these features is unique in this genus. However, Mokinastro presents the most complex sexual system within Gokinacha. Here we can see, for example, let's show you how it's complex. Let's see here. Okay. In Gokinachi, we have a Histerago with the only genus is genomonoicos. What means let's say the nomenclature of sexual system I'm following Hadford. So, in Histerago, we have 13 species with the species and the individuals. All the heads have the pistillate flowers around and the disc with bisexual flowers. Here, most of the genera in Gokinacha are hermaphroditic. It means the species produce individuals with capitulus with bisexual flowers. Look at here, you see Mokinastrum with 3 species, but I'm not sure if this species is really hermaphroditic. Why? Because most of Mokinastrum are gynodioicals. 11 species are including cyclolapse, but Mokinastrum 11 species are gynodioicals. What means we have one species and you have individuals that the flowers are pistillate, and you have individuals the flower are bisexual. But more than this, we have species polygamous dioicals. It's a very complex. Look, these 18 species presented this kind of sexual systems. Look here, we have one species, but some individuals have pistillate flowers at margin and the bisexual flowers in the disc. And individuals that all the flowers are bisexual, and individuals with flowers are female, pistillate. So this is one species producing this kind of individuals, and you have more combining here. So it's a very unusual, it's unique in the tribe. Here we can see one head with all flowers with anthers. So in this case, it was the individuals with bisexual flowers in head. And here you can see individuals with pistillate flowers. You can see only the style branches, no anthers in the head. So it's a very interesting sexual system in Mokinastro, and as I said, it's unique. This difference that we found in Mokinastro has instigated the researchers, especially Morgan, who will present us unpublished results about the tribe and the genes. So Morgan, with you now. Thank you for your attention.
Thank you, Nadia. That was an awesome presentation, very informative. I hope everybody enjoyed it. We have 5 minutes for questions. If anybody wants to ask anything, you can either ask straight on your mics, or you can put the questions in the chat.
Thank you.
Hi, Dr. Roque. Thank you for your excellent talk. I have a question for you. I was wondering if the ecology of the different genera of Guacnari that you talked about differs a lot. I know you mentioned some that were subshrubs and others that were geophytes or shrubs. Do you think that this may be correlated with different habitats that they've dispersed to and evolved in?
I think it's not, because we have Mokinastrum occurring in rock fields, in forest, but this is a question maybe Morgan will answer you, because we have discussing exactly this question, this issue in the paper, and we have some results for your question. just a little bit. Thank you. Okay, Morgan can start and you have more questions about the group. Thank you,
Yes.
everyone. Sorry, my accent in English. I hope you understand. And I hope some morphological characters can help you in your groups. Who knows? So we needed to check. We needed to looking for every feature to our groups. So that's it.
Yeah, that's a great observation, Nadja. We should look at morphology more carefully, I think, sometimes. So Morgan is going to present now some unpublished results from their work in Goknachi. And Morgan is a research botanist in the Botanical Research Institute of Texas at Fort Worth. He's been there for since 2018. And before that, he was a postdoc with Dr. Vicky Funk on the Smithsonian. And he works with lots of people that work with Asteraceae, and he's been doing a lot of work in molecular method development too. So it's with you, Morgan.
All right, thank you, everybody. Thank you, especially Nadia. Great job. Your talk was excellent. Really helped to have those photographs that help, I think, other viewers understand the micro characters that can be a little bit complicated for newcomers to this tribe. And also huge thanks to the organizers of these TICA Talks for the invitation to be here. And also I want to thank our co-authors, obviously the late Dr. Vicky Funk, my postdoctoral advisor, who introduced me to compositing and introduced me to one of the great loves of my life as a result. And then, of course, also Naji and I's colleague, Gisela Sancho, who has been a part of this work really for a long time and is another one of the world's experts really on the Goknati and Mokiniastrum. So she's not presenting today, but she is very much a part of the work that is being presented today. So anyway, this work has been a long time in the making, dating back to the start of my postdoc in 2015. I entered the Smithsonian with some amazing momentum that had been started by Jen Mandel and Vicky Funk and this team that had been working on this amazing conserved ortholog set of loci. And I thought to myself, during my dissertation research, I had started developing a new technique for comparative phylogenomics using microfluidic PCR. And so I thought to myself, what better family of plants on Earth to leverage a highly efficient molecular technique for high throughput sequencing library preparation than the most species rich family of plants on Earth, the Compositae. And so I went about looking for sort of some model groups to develop this method where I would be able to test the efficacy of these loci, both at the tribal scale and at the species scale. So I was curious about a number of different lineages, and I was really struck by the diversity of Goknati, the taxonomic foundation that had been built by Cabrera, by Nadia, by Gisela, to understand the boundaries among the genera within this tribe. And then also the inadequacy of existing molecular markers to really reconstruct these evolutionary relationships in the group. So to give you a sense of what I'm talking about, this is the phylogeny from Vicky and Nadia and Gisela's 2014 phylogeny, which used 4 molecular loci to understand the phylogenetic relationships in the group. As you can see, we have the big genera in the tribe represented within the core Goknati. Of course, as Nadia mentioned, Cyclolepis was left as Incertae sedis. However, we have strong support for monophyletic Pentaphorus, Goknatia, Anastraphia, Rictirago, Mochiniastrum, and then down here, Nicothamnus. One of the things you're probably noticing here, and which you may recall from Nagia's talk, is that in this phylogeny, Funk et al. Noticed that Gocnadia itself was non-monophyletic, but actually characterized by two clades that included a Mexican Gocnadia group, which they referred to it at the time, which has now been described as a new genus, Nahuatlia. One of the things you really notice along the backbone of this phylogeny, though, is that there's very little backbone resolution, or really no backbone resolution. The only clade that we see from any of these genera is this well-supported sister relationship between Mexican Gocnadia, aka Nahuatlia, and the Anastraphia. And so this was a little bit problematic for a number of reasons. It made it very difficult to understand evolutionary trajectories of these amazing diverse traits that are represented among members of this group, and also to understand the biogeography of this lineage, how it spread across South America into Central America and Mexico and the Caribbean. And so there were some really interesting evolutionary questions that I felt, and that we felt, we could pursue by getting our hands on a better resolved phylogeny. So this is what we set out to do. In the time since we started this work, there have been, of course, some important developments. One of which was, as Nagia mentioned, the description of Vicia, a new genus to the tribe, as well as Tehuasca, this new genus that is sister to Anastraphia in what Funk et al. 2014 referred to as the Mexican Caribbean clade. This is a monotypic genus, again, characterized by those glandular trichomes on the corolla lobes. And so if we look a little bit more closely at this phylogeny, this was based on a concatenation of ETS and ITS sequences. They used Bayesian inference to reconstruct this phylogeny. And you'll notice that Tehuasca occupies this position at the top of this cladogram, sister to Anastraphia. One of the other surprises, though, I think to those of us who've worked in Gocnadi for a while, was Panera's decision to recognize Gocnadia as a much larger genus and recognize, rather than Pentaphorus and Mochiniastrum, recognize their former sections that were described by Cabrera in the 1970s as Gocnadia section Pentaphorus and Gocnadia section Mochiniastrum. This was surprising to us for a number of reasons. One, we didn't see this relationship in Funk et al. 2014. And we also know that these are very morphologically distinct genera. They are biogeographically, the story doesn't really make a whole lot of sense. So we were curious, and one of the things we've been able to do just more recently since finding these results was kind of test this. So although we did a very large phylogenomic analysis using a large number of these conserved ortholog set loci, we were able to download sequences from Panera's 2019 study for Tehuasca, because we did not sample that in our study, to put it into the context of our larger phylogeny and determine whether or not this position is supported. And indeed, it appears that Tehuasca is indeed sister to Anastraphia. It's part of this Mexican Caribbean clade and belongs exactly where Panero hypothesized it did in his results using ITS and ETS data. However, this relationship with regard to a clade of Pentaphorus and Mochiniastrum being sister to Gocnadia is really not very strongly supported in our combined ETS and ITS data. I would call this low to medium support with a 79% maximum likelihood bootstrap. And so this was, again, surprising to us morphologically, and it was something we're we were curious about, and I'm going to present a little bit later as we look at some of the additional data. So of course, we were interested in the generic relationships in Goknati, as well as how these conserved ortholog set loci may be able to reconstruct relationships at a shallow scale, at the species scale, in this extremely interesting radiation in Mochiniastrum. Again, as Nadia mentioned, 22 species. They're characterized really easily distinguished from other genera in the group based on this paniculiform capitulescence. They're widespread throughout South America, from Argentina to Brazil to Bolivia and Venezuela. So they occupy this extremely broad and widespread distribution. And they're also characterized by having these wildly diverse sexual systems, which are unique in Goknati, in a single genus at least, having both gynomyoecious, dioecious, polygamy-dioecious, and hermaphroditic individuals, as well as having a 2 to 5 armed indiment. And these trichomes are only known also from Cyclolepis, which, as you may recall, was considered inserte setis by Funk et al. In 2014. So the objectives of this work were really twofold. First of all, we were curious whether or not, again, these conserved ortholog set loci, or at least a subset of them, could be used to reconstruct evolutionary relationships in the tribe, and then also at shallow scales within Mochiniastrum. And then if so, how do these loci inform us about trait evolution and biogeography in the tribe and in this genus? So again, I mentioned, of course, everyone is familiar with the wonderful conserved ortholog set that has proven wildly successful for reconstructing relationships in composity. These are based on a sequence capture set. And at the time, in 2015, they really didn't have, they hadn't been tested at shallow phylogenetic scales. And so this was something that would really be helpful, I think, for other synanthropologists to be able to utilize these loci. And then secondly, as part of my doctoral dissertation research, I developed a highly efficient and low cost method for high throughput sequencing library preparation using what's called microfluidic PCR. And I saw this as an interesting opportunity to leverage an existing locus set, because it's very time consuming and difficult to design these sets of loci, to design primers so that they could be used for microfluidic PCR. And so I'm going to talk just a little bit about how and why we did that. What we did was we realized that there were a number of new genomes being published across the family that could be leveraged for this purpose. And we were curious where these loci might map to on the genomes of some of these other members of the family. So we took the genome of Helianthus annuus, another unpublished genome of Centropalis pauciflorus, and then a genome from Daucus carota in, obviously not composity, but we were curious whether or not these loci might be useful in other families as well. And so we gave it a shot to see whether or not they would map to other parts of the genome in another family in the Asterales. We also, so we took 700 of these conserved ortholog set loci and we mapped them back to these 3 genomes. And what we ended up doing was taking all of those loci that did map to an orthologous region in those genomes and designing PCR primers so that we could carry out microfluidic PCR. It's important to understand, I think, what this is. So this is a method for preparing high throughput sequencing libraries that utilizes traditional PCR, but in a highly efficient and multiplexed manner. So there's an instrument that is able to carry out thousands of PCRs in a single thermal cycling program. So in about 2 to 3 hours, you can generate between 2,000 and 5,000 individual PCRs. And if you're familiar with working in the lab, typically doing something like a 96 well plate of PCRs is more standard. for comparative phylogenetics. And so the idea of being able to do 2,000 to 5,000 within 2 hours is quite remarkable. The instrument itself comes with, of course, an investment. It's an expensive instrument. It costs about $80,000. But of course, just like most sequencing facilities, there are core facilities where you can use their instruments for a moderate cost. The actual consumables that use this technology are called integrative flow circuits, but I just call them microfluidic chips. It's an easier way to think about things. And the way they're laid out is you have 48 inlets on one side of the chip and 48 inlets on the other side of the chip. On one side, you place your actual DNA extractions, your samples, and on the other side, you include your PCR primers, as well as your PCR reagents associated with your master mix. Now, when you put it into the microfluidic instrument, it pushes each of these 48 samples on one side and each of the 48 reagents on the other side in a way so that they create a matrix of, in this case, 2,300 individual reaction chambers. So you're able to do 2,300 individual PCRs at a single time. You can also multiplex the PCR primers that are included in each of those 48 reaction or sample inlets so that you can amplify up to 480 loci at a single time. So you can 10-plex, you could add 10 PCR primer pairs in a single reagent inlet for a total of 480 on one of these first generation chips. Over the past 3 years, they've developed a second generation instrument that offers a lot more flexibility for this combination of loci. And you can optimize the chip for a higher number of samples. You can optimize the chip for a higher number of loci. One of the great opportunities this offers is for $400, you can prepare up to 192 individual high throughput sequencing libraries for up to 2,400 loci. So they've improved the technology so that you can multiplex these reagent wells by 100-fold. So you can include 100 different potentially loci if you had a set of loci that's that large, like what we have for the conserved ortholog set and composite. So what this means is it brings the costs of library prep down to either $2 to $4 using the latest generation technology, or $10 per library using the original generation of this technology. So this was a great opportunity when you're thinking about trying to eventually maybe sequence 1,000 loci for the entire family. It seemed like a very efficient and low-cost way to do so, and rapid as well, since you can do this in a matter of 2 hours. So at the end of the day, you'll recall I mapped a subset of 700 of the conserved ortholog set loci back to these 3 genomes. And I ended up producing about 260 PCR primer pairs. It made sense to order a nice even round number, so I winnowed this down to a total of 240 PCR primer pairs. And then I validated these in the lab, which means I just did a regular PCR. And of those 240 PCR primer pairs, about 30% of them were successful, 78 out of the 240. And I took that 78 locus set and added to it a number of the traditional sort of usual suspect loci in composite for a total of 90 loci. So I added ETS, I added ITS, I added a bunch of chloroplast loci, NDHF, TRNLF, RBCL, PSB, TRNH, so that we would have the representation of these more traditional loci as well as these newer conserved ortholog set loci. And this is another one of the nice sort of convenient factors of microfluidic PCRs. You can easily add or remove PCR primer pairs for whatever locus you can design a primer for. So on one hand, it can be challenging to design that many primers and then see if they actually work. But once you do have working primers, you're able to easily add and subtract them from your sort of microfluidic chip as you see fit. Our taxon sampling for this, what we wanted to do was exactly mirror the taxon sampling from Funk et al. In 2014. And then we added to that number of samples from Mochiniastrum. We were able to increase the species representation in Mochiniastrum from that 2014 paper by 50% and include 80% of the species to hopefully get some really nice reconstructed relationships here. So what do we end up with when we actually looked at all of these loci? Well, although we were able to recover 78 of these conserved ortholog set loci and add them with 12 of the more traditional loci in composite, we only really had 42 that worked in a way that was able to even recover a monophyletic composite. And there are a number of reasons why some of these other loci were not successful. Because microfluidic PCR requires the same thermal cycling conditions, longer regions can not amplify as successfully as shorter regions. So sometimes with the longer regions, you get some drop off in amplification success. And we saw this in a number of those loci. We also saw a number of loci that appeared to be possibly paralogous. They were not able to recover a monophyletic composite or a monophyletic cognati. And in situations where the loci were performing like this, we thought this might be indicative of perhaps a paralogous locus. And when we were actually filtering our sequence data set, we were able to see instances of what appeared to be very likely paralogy. However, using this 42 locus set, we were able to reconstruct a remarkably well-resolved backbone for the Gochnati. As you can see, each of the, in this case, 8 genera in Gochnati are represented by different colors. And all along the backbone above the branches are red dots that correspond to high maximum likelihood bootstrap support of 90 to 100%. There are 2 branches that did not have particularly strong branch support. And these are indicated with these 2 blue arrows. And these are 2 branches that are also of note because in our Bayesian inference, we were not actually able to recover these same branches. So there's a little bit of conflict at these 2 positions in the phylogeny between our maximum likelihood and our Bayesian inference results that were only moderately supported, although they did have 68 and 74% maximum likelihood bootstrap support. This could be caused either by, in the maximum likelihood analyses, we used a partitioned analysis. However, in the Bayesian inference, we did not partition our concatenated data set. And so this difference could be due to partitioning in one data set, not partitioning in the other, or it could be due to possibly a hard polytomy. In this case, it could simply be an actual polytomy with the Mexican Caribbean clade here represented by Anastraphia nawatlia, Nicothamnus, and the clade consisting of Penniforus and Mochiniastrum sort of radiating contemporaneously as a hard polytomy. However, this is difficult to test. So we also performed a divergent staining analysis, really with the goal of trying to understand a little bit about the biogeographic history of the tribe. So we used 4 calibration points. One of them was 3 of these were secondary calibration points, which we got dates from the most recent family-wide phylogenomic study from Mandel et al. In 2019. Of course, what this meant was our calibration points were much older than the calibration points used in Funk et al. 2014. And so this pushes the dates of our divergence dating analysis much older than what was estimated in 2014. However, what we do see are two pulses of diversification. One in the tribe near the Eocene Oligocene boundary, about 33 million years ago, more or less. And this is characterized by really three important biogeographic range expansions. One in the Mexican Caribbean clade, so this long distance dispersal here of Nahuatlia and Anastraphia from the central southern Andes into Mexico and the Caribbean. And then also in both Moquiniastrum here around 33 million years ago, and in Rictarago here around 33 million years ago, a contemporaneous eastward migration from the central southern Andes into Brazil. We also saw a second pulse of diversification near the Oligocene-Miocene boundary that is characterized by this migration of what we call the densocephalum subclade in Moquiniastrum into central and southern Brazil, where we see a lot of diversification in this clade, as well as in the Anastraphia, so this migration from Mexico into the Caribbean around 26 million years ago, more or less, 24 to 26 million years ago. However, beyond this, we were really hamstrung in our ability to reconstruct a lot of the biogeographic scenarios in the tribe because of the uncertainty, especially with regard to the position of the Mexican Caribbean clade with respect to Nicothamnus and Gocnadia, which is restricted to the central Andes. So of course, while we were able to get some really strong resolution in the tribe among each of these genera, we were also, of course, curious, how well did these 42 loci perform in terms of reconstructing shallow scale relationships at the species level in Moquiniastrum? So what we find is overall, not a lot of phylogenetic resolution, unfortunately. However, we do find strong support for two clades or subclades, which we identify as the densocephalum subclade here in blue and the polymorphum subclade here in green. And as we started to look into this a little bit more closely, we saw a couple of trends emerge, and Nadia sort of indicated some of this or alluded to it in her talk, but it has to do with the morphology of the capitula in these two clades. One of these, the densocephalum clade, is entirely gynodioecious. So we have individuals that are hermaphroditic and individuals with capitula that are entirely female or with pistillate florets. However, in the polymorphum clade, we have a huge range of reproductive strategies as evidenced by it's an origin of heterogamous capitula in this subclade. So there are two origins of heterogamous capitula clearly in the Gocnadi, one in Rictarago, and then the second now in the polymorphum subclade of Moquiniastrum. So of course, again, we see a combination of both gynodioecious, hermaphroditic, as well as polygamodioecious individuals in Moquiniastrum. We're not sure if these hermaphroditic individuals, however, are truly hermaphroditic or if they've just been described as hermaphroditic because they have not been well sampled, and the only samples or collections that we have in herbaria are represented by hermaphroditic individuals, and we've just somehow missed individuals with entirely pistillate capitula that would be indicative of gynodiocy. We also see a little bit of, not much of a biogeographic signal in these two subclades. One of the subclades, the polymorphum subclade, does have a much more restricted geographical area. And so we hypothesize that possibly the evolution of these heterogamous capitula that lead to more diverse sexual systems in the polymorphum subclade may have led to or driven sympatric evolution in this subclade. Whereas in the densocephalum subclade, we have this much wider distribution, all the way up really in the Venezuela, with likely very clearly a number of instances of allopatric speciation, especially with this disjunct species Mociniastrum bolivianum in the Bolivian Andes, and Cerrado, and then eastward into central and coastal Brazil. Beyond this, though, unfortunately, our phylogenetic resolution was insufficient to be able to test some of the biogeographic hypotheses that we would have liked to, especially with regard to Isaac, your question that you had for Nadia earlier about whether or not particular traits are correlated with particular habitats. And it's really a shame, because this was something we were really hoping to dig into in Mociniastrum, because they really do occupy such a wide variety of habitats, from the very dry or seasonally dry Caatinga in Brazil to the Cerrado, to these campos rupestres, these rocky fields, and the Atlantic forest. So there's really an untapped opportunity here, because these loci were not sufficiently variable. So with that, those are kind of the big takeaways that we have. Among our objectives, we were curious whether or not these conserved ortholog set loci would be able to reconstruct evolutionary relationships. And they really do, especially in the genus level. So we have very strong support for well-resolved backbone phylogeny in the tribe. This is something that is very special. It allows us to really dig into a little bit of morphological trait evolution, and we begin to see some really interesting evolution of things like, in particular, heterogamous capitula restricted to just a single subclade of Mociniastrum and Rictarago. We also see that, unfortunately, we aren't able to test some of the hypotheses that we had about trait evolution and biogeography in Mociniastrum as much as we would have liked to. And that's just, unfortunately, going to have to be tabled until I think we can include a larger representation of these conserved ortholog set loci. So some of the conclusions and takeaways and future directions where we hope to go in the near future with this data is, first of all, we're very excited about these new results. However, we do need to sample 2 of these new genera that have just recently been described in the last 2 years, including Vicia, which would be fantastic if this can be rediscovered growing in the wild. It's very likely extinct. It's known only from a handful of collections, restricted to very highly urbanized environments. So if we can rediscover this actually growing, it would be fantastic to include it in our analyses, as well as Tahuasca. Of course, we were only able to include sequences from ITS and ETS that we had from Panera's work on Chenbang. And so including that in a broader representation of loci would be a big help. Importantly, what we do find is very strong support for monophyletic Gochnati that includes now 10 genera. And one of those is Cyclolepis, which was formerly Incertae setus. So it's great to actually have a position for Cyclolepis in the phylogeny and included in this tribe. Of course, we do see some interesting morphological trait evolution going on with regard to the evolution of heterogamous capitula. It's very clear that in Mociniastrum, we have a model system to test the evolution of sexual systems, complex sexual systems, and possibly to test hypotheses regarding the evolution of Diocese. Unfortunately, we just don't have that resolution to take it further, but it's clear that the evolution of these heterogamous capitula did likely trigger cladogenesis in Mociniastrum, which is really, really cool. We do find that microfluidics is an effective method for fresh herbarium tissue, fresh and preserved herbarium tissues. Of our 56 samples that we tested from herbaria, 48 of them, or almost 90%, were successful from tissue up to 30 years old. So this was exciting. And I think one of the challenges is just going to be being able to design PCR primers that are amplifiable under the same thermal cycler conditions. This is a major limitation of microfluidics, and it's one of the trade-offs. Of course, you're able to develop high throughput sequencing libraries very cheaply and very quickly, but you're not going to be able to amplify anywhere near or sequence anywhere near the same number of loci that you get with in situ hybridization. So we have improved phylogenetic resolution, but it's limited. And obviously going forward, I think we've got some really exciting hypotheses to test. I think this would be a great study for a PhD student to take on, to dig into Mochiniastrum a little bit more closely using in situ hybridization of these conserved ortholog set loci to hopefully reconstruct these relationships more closely and understand what is going on with sexual system evolution as well as biogeography. So with that, gracias, obrigado, and thank you to a huge number of people that have helped guide this project along from day one, almost 6 years ago. And with that, I hope we have time to take questions. Thanks.
Thank you, Morgan. That was a great talk. If anybody has questions, we do have time. You can, again, speak, say your questions out loud or put them in the chat. Well, since nobody has questions for now, Morgan, you should write a small article about the microfluidics PCR to include in the next volume of Capitulum.
I could do that, actually. Yeah, I think. I'm really busy over the next 3 months, but I could maybe do that. The next deadline is October, is that correct?
I think Mauricio would know better than I do.
Let me think on it, and let me give it some thought.
That will be awesome if you could do that. You will need to get us a first draft by the end of September. So yeah, that will be awesome if you could produce such a thing. I think it's a very interesting approach to getting data. So
we have 2 questions on the chat. So Ed Schilling is asking, can you identify a small number of loci that might be checked for a larger number of samples by traditional Sanger sequencing?
Yeah, absolutely. That was one of the things that I did when I was designing the overall set. Was I did a large literature review of basically every locus that has been used in composite over the last 30 years to try and make sure I was including as many possible loci as I could. And some of them were a little bit obscure. And among those, there were a couple that actually performed pretty well. I can't remember the names off the top of my head, but I think a couple of them were from the Bruce Baldwin 2002 Helianthe Alliance paper in systematic botany, I want to say, that haven't been used very broadly in a number of other studies, but it was easy to develop primers for. They weren't too long, so they were amenable to the microfluidic technique. I think one of the challenges for these loci that you're able to design primers for is that having that requirement of being able to design PCR primers leads you to kind of be selecting for loci that are already a little bit more conserved than what you might get using in situ hybridization, where you get more valuable or more variable loci that have these off-targeted sequences near the boundary of those targets that you're looking for. and they're evolving more rapidly, and you get more variability. So in this case, I think it may be likely that a lot of the loci I was able to retain for microfluidic PCR were just constrained by sort of a self-selected conservatism among the loci, if that makes sense. I am really excited to just kind of continue pushing this forward as new loci become refined, the data sets become, I think, better and more broadly tested. We're going to be able to really get data sets that are more inclusive, flexible. And I think in a big way, it's a nice opportunity to hopefully continually evolve these loci in a way that can work with larger and smaller groups. And I think use and combine both approaches. So in this case, we were looking at really two questions of different phylogenetic scales. And so being able to use a much larger number of samples, but only including a smaller number of loci, although still a very large number of loci compared to traditional comparative phylogenetics, is sort of just that middle ground that we might be able to bargain with in terms of exploring massive phylogenetic space like we have in composite. So somehow finding a middle ground between using techniques that might be more labor and cost intensive and those that might be more rapid and cheaper and affordable. That's my hope anyway. And I know as a community, we're going to continue to see these loci just get better and better. Anyway, it's a long answer and to your question, and then a lot of extra info.
We have a second question on the chat, Morgan, about the percentage of informative characters and sequence alignment.
Good question. We used MAFT for sequence alignment. So of course, we are, any phylogeny is in a way beholden to the alignment. So that's always something I'm very aware of. I did compare MAFT and muscle alignments. So I first prepared back in 2018 this data set using muscle. And as I started to refine the data set and remove loci that appeared to be paralogous, I later ended up using MAFT, honestly, as a matter of simplicity, because the MAFT web application is very easy to use and quick. But also because it was an opportunity to compare muscle and MAFT. I ran the muscle alignments locally on my computer, and then I ran the MAFT alignments in the cloud. And so being able to compare them was nice. We didn't really see big differences in the topology between the muscle and the MAFT phylogenies. Percentage of informative characters was between, depending on the locus, anywhere from like 1.5% to 14% informative. So there was a wide range. And I am curious about pulling out some of these loci to figure out which ones might be more informative at shallower scales, more variable. I honestly think we're in a perfect point right now as a composite community to do a meta-analysis maybe from a lot of these published locus data sets to determine maybe some of the subsets of loci that are more informative for some rather than others. I've been excited about that for a long time. Finally being able to meta-analyze aggregated data sets from the past like 6 years of phylogenomics. So great question.
The meta-analysis sounds very interesting. I would definitely be interested in seeing the results from that. Jennifer just put a very long question on the chat. So Jennifer, if you want to say the question out loud, or Morgan and Nadia can read it if they want to.
I'll let them read it because there's a lot of detail, and plus they're drilling outside my door.
Okay.
Yeah.
So the question is, I'll go ahead and read it for everybody. In our limited sampling, the 2019 PNAS paper, the Cyclolepis was sister to the Wunderliche. And in these results that I presented, it was sister to Gochnati. It's part of Gochnati. And so, Jen, you said you just looked at support, I assume from the 2019
Yeah,
paper,
yeah.
and it was very low.
It's in the
that
60s.
sister.
Yeah, it's in the 60s for that clade for the maximum likelihood approach.
And this is something that was also consistent in the 2014 paper as well. You had the, I believe, nuclear loci in Funk et al. 2014 were placing Cyclolepis very weakly supported as sister to the rest of Gochnati, but in the chloroplast data set, it was also very weakly sister to Wunderliche. And this is why they chose to leave it as incertae sedis, even though I think in the combined nuclear plus plastid data set, Cyclolepis was still weakly supported as sister to Wunderliche. They chose to leave it as incertae sedis because morphologically it really belongs in Gochnati. I mean, for Cyclolepis to have not been included in Gochnati was a surprise from that 2014 paper. And so because the branch support was so low, it does raise a good question though, whether or not this is the result of possibly ancient hybridization, especially since that chloroplast data was so wildly different. And in the results presented here, there's definitely a bias toward nuclear loci, although we do have quite a few chloroplast loci that I was able to include from traditional marker sets. It's a great question. And it's one I'm curious to see play out over the next few years as we hopefully, by having these data sets, can spark some curiosity in students and other researchers to take on the type of ecological work that might help us answer that question, to understand really just how common maybe hybridization might be in compositae, in other groups, how to look for signatures of hybridization in phylogenetic data sets. It's a great question. I'll be curious to see that. And it gets to other questions that I'm really curious about in terms of, I'm gonna leave that as something we don't have time to talk about, but great question. It's possible. The support in this study though really confidently places Cyclolepis within Gochnati without a question, including independent analyses of our outgroups and the rest of the composite. So I'll let Nadia maybe chip in.
Jennifer, I didn't see this the last results from the phylogeny. I couldn't read. I was traveling, and I did see the last version. So for me, it's new. What Morgan presented is the last version. It's new for me. So morphologically talking, I think Cyclolepis should stay inside Gochnatoid. For me, it makes sense. But now I need to, we need to read the paper and see the discussion. But a good question. It's a long time, it's incertae sedis. And I'm happy we finished this question, I hope.
Cool, thank you.
Well, if we don't have any more questions, I think we can finish the session. Thank you, Nadia and Morgan. It was a pleasure to have you both here today to talk about this very interesting and weird group of plants. Our next session is going to be in September, and we're going to have Dr. Gustavo Hayden talking about Baccharis. and it's going to be another very good talk probably. We still have some space for speakers in October and November. So if you are interested, please send an email to the TICATalks email. And we also have a form that you can use to register if you're interested in speaking, that is available on the TICA website at composite.org. This talk is being recorded and is going to be available on YouTube later this week. So make sure to check the YouTube channel. And if you've registered for this talk and you got the email from the TICATalks email with the Zoom link, you don't need to register again. You're going to receive the emails every month with the announcements and the Zoom links. So you don't have to worry about registering again. Thank you so much, and hope to see you again next month. And if anybody has any other comments, we still have time to.
Thank you. Thank you to the TICA team for organizing this. And if anybody does have questions, I'd be happy to stay on or send an email to Nadia and I. I know we'd be, and look for our paper, which should be coming out here soon to have it now that it's finally completed.
Thank you,
thank
everyone.
you, Nadia.
See
Thank you, Nadia, and thank you,
you.
Morgan. And remember to join TICA if you haven't done so. Okay?
Okay.